Hi, this is Hsuan Lin. I'm from Parallel from Chemistry Department. Today I'm going to talk about using two small molecules to control protein concentration precisely in living system. So relative abundance of reactants in a flask can lead to testable and predictable outcomes. However, a similar understanding in cell signaling is limited. Um, we hypothesize that the protein concentration controls the dynamic as well as thermodynamic of cell signaling. However, the rate limiting rate limiting step for analyzing this type of problem is a lack of tools to precisely and conditionally control protein concentration. So researchers have been using small molecules to control protein concentration. One of the most successful methods is by fusing so-called destabilization domain to your protein of interest. For example, Wendel's group, they fuse a mutant FKBP to their protein of interest, and then when they add this small molecule called rapamycin, it will go bind to the FKBP and thus stabilize the fusion protein. So the fusion protein or the protein of interest will be functional. However, despite the successful application of their research, they result in a fusion of a tag to your protein of interest. For some proteins, this tag might be well tolerated. However, for some proteins, it might affect its natural behavior. So a tag-free technology will be more suitable in this case. So here in Parallel, we make use of a technology called split ubiquitin. Ubiquitin is a 76 amino acid protein that's reside in cells, either free or covalently linked to other proteins. While it's being made, it will be cleaved at the junction of the fusion and thus release a free ubiquitin by a dedicated ubiquitin hydrolase. Split ubiquitin was introduced by Johnson-Warshawski in 1994 when they were developing tools for monitoring protein-protein interactions. In short, ubiquitin was split into N and C terminus, corresponding to 1 to 37, 35 to 76 amino acids, respectively. When there's a dimerization signal brought those two fragments to each other, they will become folded again, and the hydrolase will come in and cleave at the junction of ubiquitin and your protein of interest. So we developed this system called traceless protein shielding and a T-shell system. In this system, we made use of the mutant FKBP as our destabilization domain. We fuse that to the C terminus of ubiquitin and then followed by our protein of interest. Another construct of this system is FRB fused to the N terminus of ubiquitin. When we add rapamycin into the cells, it will go bind to the mutant FKBP, thus stabilizing it, and then it will recruit another piece. So ubiquitin N and C terminus will become folded again and recognized by the hydrolase in the cells, thus releasing our protein of interest in its intact form. So we also put the T-shell system under the effect of cation advanced expression system. In this system, we can add another small molecule called doxycycline into the cells. Doxycycline will bind to the cation transactivator, and then the resulting complex will bind to the sequence that drive the production. Of our system. So more of the doxycycline we add to the cells, more of the system will be expressed. And then when we add more of the rapamycin, more GFP, which is our model protein in this case, more of the green fluorescence protein will be released. So this is the result of our experiment when we have 9 different concentration combination of 2 small molecules. When we have more of them, more of the green fluorescence protein will be released, and then we monitor this fluorescence using a machine called flow cytometry to measure the live fluorescence coming out from the cells. So we were able to achieve a 130-fold dynamic range compared to the background level. The plasmid, which is the circular DNA that's encoded the TCO system is available on Etching, which is a non-profit plasma sharing company. Thank you for your attention.